Dear students, this is our 15th lecture on medicinal chemistry, and today we will start with a new chapter that is on antiviral agents. This will be our first lecture on antiviral agents. Antiviral agents or antiviral drugs, basically these are a class of medication which is used to treat viral infections in our body. The majority of antiviral drugs or agents, they target specific viruses in our body, whereas there are many antiviral agents which will be called as broad spectrum antivirals, because they will be effective against a wide range of viruses. Because viruses, they are of many types, and most of the common viruses which we know are very common these days. One is HIV, human immunodeficiency virus, then herpes simplex virus, which is called as HSV, then hepatitis B, hepatitis C, then varicella zoster virus, then some respiratory syncytial virus, then influenza A and influenza B viruses. So these are the category of different types of viruses which causes different types of diseases in our body, and their mechanism of action for most of these viruses is same. So there may be some antiviral compounds which may be effective against many of these or all of these. Such type of antiviral compounds, they are called as broad spectrum antivirals. Now, antiviral drugs, these are basically one class of antimicrobial drugs. Antimicrobial drugs is a larger group of medicine which includes antibiotics also. Antibiotics, they are also called as antibacterials. Antibiotics, antifungal agents, and antiparasitic drugs. So all these drugs together, along with antiviral compounds, they are called as antimicrobial drugs. Then antiviral drugs may be based on monoclonal antibodies also. Antiviral drugs, they are different from virucides. Virucide, or this is called as viricide or biocidal agents. The difference between an antiviral drug and virucide is that a virucide will deactivate or destroy the virus, whereas antiviral drug will inactivate the replication of the virus. So this is the difference between virucide and antiviral drug. Virucide will either deactivate or destroy the virus, whereas antiviral drug will stop the replication of the virus. The replication of the virus will cause different types of diseases in the body. So there are some natural virucides also which can deactivate or destroy the viruses, and these are produced by some plants such as eucalyptus and Australian tea trees. So they give us natural virucides which can deactivate or destroy the viruses. Now, what are viruses? Viruses are intracellular parasites, and what they lack is they don't have enzymes. And we know that enzymes, they have RNA or DNA, which is used for replication and for making structural component. So because the viruses, they lack enzymes, therefore to replicate or for making structural component, they use the host cell in our body to perform these activities. So that is why you can say that virus is a parasite on our cell in the body. So basically what happens is that viruses they literally hijack the host cell, and they will take over its structural components or machinery, and they will start replicating the viruses to have more number of viruses. Now when we use antiviral drug, the challenge before us is that that drug which we are using as the antiviral drug, that should kill only virus, and it should not destroy our body cells, otherwise it will not be effective. So there need to be selectivity in the antiviral agent which we are going to use. So therefore the drugs which are targeted against viruses, they must be selectively toxic to inhibit the viral particles without adversely affecting the host cell. Only then the antiviral agent will be effective for treating that disease which is caused because of the virus. Now, virus consists of a genome, which is nucleic acid, surrounded by a protein coating called as capsid. The nucleic acid which is present in the virus may either be DNA or it may be RNA. Both are not present as genome. So either there will be DNA or it will be RNA. And in addition, the most animal viruses, they will have an envelope which is called as lipid envelope, which is having protein spike receptors which will help the virus to attach on the host cell surface. So this is the structure of different types of viruses. The antiviral drugs, what they do is they will inhibit the viral replication. So as I have told you, this is how we can differentiate between antiviral drug and a viruside. Viruside will deactivate or destroy the viral, whereas destroy the virus, whereas antiviral drug will inhibit the viral replication. So antiviral drugs, they will use two approaches. One is they can directly target the virus, or they can target the virus through host cell. And in the host cell, they can act, antiviral agents, they can act in different manners. They can act as inhibitors of virus attachment or entry. Then they can be used as uncoating inhibitors. Then they can be used as inhibitors of viral replication. Then these antiviral agents, they can inhibit the protein synthesis in the virus, or they can inhibit the viral assembly. And last is that these antiviral agents, they can inhibit the release of viruses in the body. Now as far as this mechanism of replication process is concerned, say this is our host cell in which this virus will attack, and it will go through different stages for replication. Now for example, this is a virus which is having a genome which may be of RNA or DNA, as I have told you, and this is surrounded by capsid and then by the lipid envelope. Now through these spikes, it will enter into the host cell, and in the first step, virus will fuse with the host cell, and then in the second step, it will penetrate through the host cell, and then this coating will be destroyed. So uncoating will be the next step, and uncoating will release this genome RNA or DNA strand into the host cell. So now we are having either viral RNA or DNA, whatsoever genome it is. Then this viral RNA will undergo reverse transcription, and transcription will lead to the formation of double helical viral DNA. Then this will enter the nucleus and undergo integrase, and in reaction with host DNA, the viral genomic RNA will be produced, and then viral messenger RNA will be produced. So next step is integration of host DNA with viral DNA, as I have told you. Then transcription will take place, and then viral messenger RNA will be there, and this will lead to the again formation of through translation the virus replication will start, and after the replication of virus, packaging and assembly then release into the virus will be released into the body through this host cell. So this is the whole process of replication of uh, virus. Now, what antiviral agents they do is that, as I've told you, there are different types of antiviral agents. First category is entry or fusion inhibitors. So such type of antiviral agents, they will act on the virus at this stage, and they will inhibit the entry or fusion of the viruses to the host cell. So if this antiviral agent is successful, then the whole process will not take place. The virus will stop at this stage only, right? So, and if this is not successful, then virus will undergo uncoating, and then to stop that uncoating also we can use ion channel blockers, which will try to inhibit the uncoating or try to inhibit the uncoating of this virus. Then as we know that after the uncoating, this RNA is going through reverse transcription phase. So here we can use reverse transcriptase inhibitors. So these antiviral agents, they will not allow the virus to undergo this step. So when this step does not happen, that means the further replication of virus will stop. And if even this is not successful, then we know that the next step is integration of host DNA with viral DNA. Then we can use integrase inhibitors, which can stop this process, and then also further replication of viruses cannot take place. But if this is also not successful, then we can use protease inhibitors in the next step, which can stop this packaging and assembly. Then in the last, if all these steps they are not successful, then we can stop this budding or viral release through neuraminidase inhibitors. These can stop the viral release from the host cell to the body. So this is how, through different types of antiviral agents, we can stop the replication process of virus to stop the host cell from releasing an infectious virus in our body. Now we can discuss the various types of antiviral agents which can be used. As I've told you, first is inhibitors of virus attachment, fusion, and entry inhibitors. So these drugs, they what they do is they target host receptors, core receptors, or viral spike proteins. So they will interact with these receptors, core receptors, or spike proteins, and they will inhibit the action of any of these or all of these. So when this drug will inhibit the attachment and virus entry, prevent all subsequent steps of viral replication cycle, as I have already told you. So what this will do, this will permit the clearing of virus by the host immune system at the beginning of the virus replication. And these types of inhibitors, they target mainly the enveloped viruses which are having the lipid membrane present with the spike proteins. So only those types of viruses, they are affected by these types of inhibitor drugs. Example is maraviroc and enfuviretide. This is a natural product which is isolated from different types of cell membranes, and this is also uh, used as inhibitor of viral attachment or fusion. So what this will do, this will bind the GP41 of the viral envelope and inhibit the viral fusion with the host cell. Whereas this maraviroc, this binds CCR5 of the host cell receptor, and this will block the attachment of the viral to the host cell. So this is how they will act and inhibit the attachment of the virus or fusion of the virus at the cell membrane of our host cell. Then is inhibition of viral uncoating in the host cell. So these types of drugs, they will prevent uncoating and release of the viral genome in the cell. And most commonly used is amantadine. This is the structure of the compound, or amantadine. So these are derivatives of adamantane. So this is primary amine, whereas which is directly attached to adamantane molecule, whereas this is 2-ethyl amino group which is attached to adamantane, and this is amantadine. This one is a narrow spectrum drug which works against only influenza A viruses. And this amantadine, this blocks M2 ion channel function and thereby prevents acidification, dissociation, and uncoating of the influenza A viruses. Then inhibitors of viral replication, two types of antiviral agents, they can be used. One is polymerase transcript inhibitors, other is reverse transcriptase inhibitors. Polymerase transcriptase inhibitors, they will target DNA or RNA polymerase to inhibit the DNA oblique RNA replication in the host cell, to inhibit the activity of the virus in the host cell. So here the viral DNA polymerase inhibitors like acyclovir, hydroxyridine, and tenofovir, this is the structure for all these three. They can be used as polymerase transcriptase inhibitors. So here you can see these are mainly bases or alkaloids which are used as polymerase transcriptase inhibitors. Then reverse transcriptase inhibitors, they will target the reverse transcriptase enzyme, which will inhibit the synthesis of DNA from RNA. So these different two types of drugs which can be used under reverse transcriptase inhibitor category. That is, these can be based on nucleotide or nucleoside analogs, or they may be non-nucleoside uh, non-nucleoside analogs. So nucleotide or nucleoside analogs, they will compete with regular nucleotide or nucleoside which is present in the host cell, and they will insert themselves into the growing nucleic acid chain. So basically, instead of a regular nucleoside or nucleotide, now we will have the antiviral agent as a nucleoside or nucleotide into the process of growing nucleic chain. So that means the replication of DNA from RNA, that will stop permanently. So this is the mechanism of action in case of use of nucleotide or nucleoside analogs as antiviral agents. Then this nevirapine, this alkaloidal molecule can be used as reverse transcriptase inhibitor. This is non-nucleoside inhibitor, and this will non-competitively bind the polymerase or reverse transcriptase enzyme. So because of this binding of this nevirapine, the process of replication will stop. Then is integrase inhibitors. These antiviral drugs, they will provide prevent the binding of viral genome to the host gene, and we know that binding of these two, viral and host genome, is essential for these viruses for integration process. And most commonly used drug is raltegravir and dolutegravir. This is the structure for these. These are again heterocyclic compounds which can effectively act as inhibitors to integration of viral with the host gene. Then inhibition of viral protein synthesis. So these antiviral drugs, they will inhibit the protein synthesis by an antisense mechanism. So basically, antisense antiviral drug is a short synthetic nucleic acid strand that complements the specific part of messenger RNA. And this messenger RNA, what it will do, which will bind this antisense nucleic acid synthetic strand will bind to mRNA and prevent the protein being translated. So basically, this will stop the translation of protein. That is why inhibition of protein synthesis will take place. And most commonly used antiviral drug is fomiviricin. So you can see all these are the synthetic strands of nucleic acids. Then is protease inhibitor, that is inhibitors of viral assembly. These types of antiviral drugs, they will inhibit the viral assembly category. The most commonly used, there are many antiviral drugs: ritonavir, atazanavir, darunavir, sequinavir, indinavir, dalfinavir. All these, you can see, they have almost similar type of structure, having different types of amide present, polyamidic structures they are having. So they can act as inhibitors of viral assembly. Basically, what they will do, they will block the active sites of specific protease enzyme. And due to this, polyproteins will not get prolifically cut into their final proteins, and that is how protease inhibition will take place. And different types of protease inhibitors, they will act against different types of viruses. For example, these squinavir, tenavir, indinavir, and alphanavir, they will act as inhibitors against HIV viruses. So different types of antiviral drugs, they will be used for specific type of viruses. Then is inhibitors of viral release, neuroaminidase inhibitors. These antiviral drugs, they will inhibit the last step, that is release of viruses from the host cell to the body. So, anti-influenza or anti-COVID-19 drugs like oseltamivir, which you must have heard many times during COVID periods, what they do is they will block neuroaminidase, which is this is required to release a new virus from a host cell into the body. So, if these antiviral drugs, they will block this neuroaminidase, that means the replicated or assembly of the viruses, they will not be released in the body. So beside these drugs that target viruses, some drugs they have been developed, they will act as immunomodulators like nitazoxanide, which will interfere with host-regulated pathways of viral replication, that is amplification of type 1 interferon pathways, and cytoplasmic RNA sensing. Similarly, ivermectin, which you might have heard many times about in the COVID period also, this inhibits the nuclear import of the host and viral proteins. So these are the structures of different types of antiviral agents which act as inhibitors of viral release. Oseltamivir, nitazoxanide, ivermectin. So all these have different types of structures, but their mechanism of action is same, that they will inhibit the neuroaminidase activity. So this is the table which tells us that what type of drugs they are used for what type of inhibition in the different types of viruses. For example, inhibitors of viral attachment entry for COVID-19, the most commonly used drug is hydroxychloroquine and chloroquine. So these can be used for COVID-19, whereas enfuvirtide anti-HIV, maraviroc anti-HIV. So their mechanism of action is that they will make increase in the endosomal pH, which is needed for the viral or cell fusion. Then is uncoating inhibitors, preventing the release of the genome. Amantadine, as I have told you, and ramentadine, which is used against influenza A. This I have already told you that they will inhibit the M2 ion channel, preventing pH-dependent dissociation of viral proteins. So therefore, release of nucleic acid to the host cell will be prevented. Then inhibition of viral replication, remdesivir for COVID-19, favipiravir for COVID-19, foscarnet for HSV, acyclovir and ganciclovir. for herpes simplex virus, ribavirin for RSV, dolutegravir, and dolutegravir for HIV, zadovudine for HIV, lamivudine for HIV and HBV, nevirapine and efruxifermin for HIV. So different types of drugs they can be used for inhibition of viral replication, and the mechanism of action of these drugs is RNA independent RNA polymerase inhibitors. So these are analogs of adenosine nucleotide. Similarly, viral protein synthesis can be inhibited through these drugs in different types of viruses. Then inhibitors of viral assembly, ritonavir, lopinavir for COVID-19, then HCV, boceprevir, atazanavir, HIV. They will inhibit the synthesis of the protease enzyme. Then inhibitors of viral release, oseltamivir for influenza and COVID-19, zanamivir for influenza. What they will do is they will block neuroimmunities, which is required for the release of a new virus. So these are some general antiviral drugs which are used for inhibition of different types of steps in the antiviral mechanism. So these are the references which have been used for preparing this lecture. Thank you very much.